Hey everyone, this is Dr. Marie McNeely, welcoming you to episode 693 of People Behind the Science. Today, we'll be chatting with our guest, Dr. Adriana San Miguel. And listeners, I appreciate all of you joining me today, and I want to give a special shout out to everyone who has rated and reviewed our podcast on your favorite platforms. This helps new listeners get excited about science and, of course, the people who are making fantastic research discoveries today. So if you haven't rated or reviewed our show yet, we would love to hear from you. And listeners, I hope you're ready to meet another one of our exceptional people behind the science after a quick message from this episode's sponsor.
For 45 years, Lampire Biological Laboratories has been a key player in the life science industry. We are an international biotech company that produces biological reagents. For a complete listing of products, you can visit our website at lampire.com or call 215-795-2838.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Adriana San Miguel. So, Adriana, thank you so much for joining us on the show today.
Thank you so much for inviting me, Marie.
Well, we are excited to have you with us, and I'm looking forward to telling our listeners a little bit more about your background. So, listeners, Adriana is an assistant professor in the Department of Chemical and Biomolecular Engineering at North Carolina State University. She received her bachelor's degree in chemical engineering from ITESM, a technological institute in Monterrey, Mexico. As an undergraduate, Adriana received the Frisa Entrepreneurship Award from ITESM, as well as the Craig P. Dunn Award for Social Innovations in Entrepreneurship from the San Diego State University Venture Challenge. Now, prior to starting graduate school, she worked in the cement and water treatment industries for a few years, and she was awarded her PhD in chemical engineering from Georgia Tech. And during her PhD, Adriana was recognized with the Exemplary Academic Achievement Award and the Ziegler Award for Best PhD Thesis Proposal from the School of Chemical and Biomolecular Engineering at Georgia Tech. Afterwards, Adriana worked as a postdoctoral fellow at Georgia Tech and also at the Dana-Farber Cancer Institute. As a postdoc, she received an NIH K99 Pathway to Independence Award before joining the faculty there at NCSU. And today, Adriana, we're excited to get to know you both as a scientist, of course, but also as a person. So can you start by telling us what you like to do when you're not doing science?
Well, I'm generally pretty busy. Aside from being a scientist, I'm also a mom of 2 young kids. So that does not leave me a lot of time. But when I do have time or I make time, I really enjoy working out. I used to be a runner, and now I tend to swim a lot. So that's something I really enjoy. It makes me happy and it just gets rid of stress.
Oh, wonderful. I think that is a great way to burn off some of your sort of nervous energy there. What are the kids' favorite activities these days?
Well, they're very young, so they go to daycare most of the day. They're almost 2 and almost 4. So,
aw. Well, it's great to hear a little bit more about your life outside of science, but I'd love to talk about your research as well. So, Adriana, how do you describe your work to people who aren't in your specific field or maybe aren't in science at all?
So what we do in my research group is we use this little model organism. It's a small nematode called C. elegans. And what we try to do with this little nematode is we try to understand processes such as aging, neurodegeneration, and stress. And I'm actually an engineer, and we use engineering tools to try to make these studies faster than traditional techniques and also more quantitative. And what that means is that we're able to really numerically determine what's changing in these animals as they age or as they degenerate their neurons, for instance.
Oh, wonderful. And I must admit, C. elegans are near and dear to my heart. One of my first research projects ever was on these, what I think are adorable little worms. So
they are adorable and it's very fun to work with them.
Definitely. Well, I'm excited to chat about your work as we go through our conversation today, but I'd love to talk about motivation at the beginning of our show. I know as a scientist, it can be difficult to stay motivated and inspired because the work that you do is difficult. So for you, do you have a favorite quote or a saying or something that really keeps you motivated?
I don't think I have a quote or a saying. But yeah, I agree. Most of the times research projects tend to be long, right? And so staying motivated through years that it takes to finalize a research project can be hard. And this is true for students or PIs or everybody. And I think really what motivates me more is just seeing the students grow through the process, right? And when you find something completely unexpected, those are really fun and puzzling things to go after. And then that's what leads you to new projects and new research ideas. So I think the fact that we can just investigate new things and just see my students grow, I think that's what keeps me going.
Wonderful. I love that sort of excitement of discovery and almost seeing that through new eyes every time you have new students come through the lab is absolutely wonderful.
Oh, yeah, it is.
So for you, Adriana, when you think back around your own career, your own life, are there particular people who really inspired you or motivated you or maybe encouraged you to pursue science that you look up to?
For sure. I think mentors are super important, and I believe that myself. I think I have a few people in mind. Of course, my PhD advisor, he was just someone that I encountered that was just really passionate about science and just really interested in the fundamentals of what we were studying at the time. And just seeing that in somebody else sort of motivated me a lot. And I just felt this new appreciation for the beauty of science itself. And then the other person that's been very important in my career has been my postdoc advisor. And she was just an incredible cheerleader for me. She was always, motivating me and telling me I could do things, even though I didn't think I could at the time. And I really think that I am here today because of them. Also, during my PhD, I took a few classes that were not in my area of expertise. And some of the professors I took classes with really inspired me, and sort of the knowledge that I learned from those classes opened a new world to me, which is biology. And that's how I'm doing what I do now.
Absolutely. And I love how your work really brings together the biology with the engineering. And this is sort of a relatively unique approach to science. So can you talk a little bit about how you got here, Adriana? Where did it all begin for you and how did you start on this path?
Actually, I like to talk about this a little bit because I think my path has not been very traditional or very direct. Mm-hmm. It's been a little bit of a winding road to get to where I am. And I think I like to tell that story because there's other people that can feel motivated by that, right? There's not always a direct path or a direct number of steps you need to take to get to be a professor in a specific area. So I'm originally from Mexico and I did my undergraduate studies back home in chemical engineering. And then after graduating, I actually worked in industry for a couple of years. But when I was working, I always knew I wanted to go to grad school because I was interested in science and I just wanted a higher degree, although maybe I didn't know a whole lot about what research meant at the time. So then I moved to the US to pursue a PhD in chemical and biomolecular engineering. And that's where I learned about all the different types of research you could do in chemical engineering. And I picked something very different from what I was originally thinking I was going to do. I was originally thinking I would go and do research on nanotechnology. And I kind of changed my mind after learning about other types of research. And so then I did my PhD, which was actually focused on colloidal systems and stimulus responsive emulsions and capsules, which does not have a lot of bio in it, to be honest. But then, as I mentioned earlier, I did a minor in biology. I took some biology classes. And the reason for that was I don't know a whole lot of biology, so let me learn some. And then I just really fell in love with biology and genes and proteins. And so after that, I did a postdoc where we combined engineering with C. elegans, so something similar to what I'm working on today. And I never thought I was going to be a professor, or that was not my goal when I started grad school. But I guess I started getting more and more interested in the science and the mentorship aspect of it. So I said, I'll give it a shot. And so here I am.
Oh, that is wonderful. And I'm so glad you shared your story because I think you're absolutely right. These winding paths are, I think, a little bit more common than people realize. I think in science, you sort of perceive there's these three steps. You go to graduate school, you get a postdoc, you get a faculty job. And I think there's so many other ways to get to this same position, which is absolutely wonderful. And your industry position after undergrad, I think is fascinating. You actually worked in the cement and water treatment industries. Can you tell us what this transition was like going from undergrad into this kind of an industry role?
Right. That was great in some aspects and not so great in other aspects. Mm-hmm. So the water treatment industry, I worked there as an intern when I was doing my undergrad. So that was an easy transition. But then after that, I actually moved to this little town in the middle of nowhere back in Mexico, where there was this big cement producing plant. And that was kind of a little bit of a shock for multiple reasons. One, because I was living in a very small town coming from a big city. And then the other one is the cement industry is mainly a male dominated industry, right? And I was a young engineer woman. So that was a little bit of a culture shock. But people were very, very nice to me. I just did not really enjoy that terribly. And so the actual work that's done. So at that point, I decided to apply to grad school, although that was in my mind the entire time.
Definitely. So was it difficult then the next transition into graduate school after you had sort of been working a regular job?
Right. Well, I think that the difficult part there was moving to a different country. To me, that was the difficult part, and sort of leaving your family and all your friends behind. So that was a little bit hard, but just going back to school wasn't terribly hard. Starting research, I hadn't done a lot of research in my undergrad. So starting research and really figuring out what that meant. And there's a lot of resilience you have to build and be okay with experiments failing multiple times, all these things students learn as they go. That was a little bit hard, but it's just what you do in your PhD. And thankfully, with a really good advisor like I had, it became exciting and easy to deal with.
Absolutely. I think having that right mentor can make all the difference in your graduate school experience. And it sounds like, for the most part, everything went great for you. You made it there to North Carolina State University on the faculty now, and you've got some amazing projects going on in the lab that I'd love to talk about. So, Adriana, do you have a favorite one in the works at the moment that you want to share with us?
So we have multiple ones. I think one that I'm excited about is trying to understand how neurons change shape as they age or they degenerate. So we've been working for a little while on using convolutional neural networks, so a type of machine learning approach where we're using these tools to analyze images. And these images are images of neurons in living nematodes. And the reason we need to use these tools is because the specific neurons we're looking at have very complex patterns or morphology. And so really determining if there's a significant change in their structure or if it shows any specific features indicative of damage is very hard, especially if you want to do it quantitatively. Like, for example, these neurons start to show these little bubbles as they age, and you can also see those with other types of stressors that induce degeneration. And a single neuron of the type we're studying can show anywhere from 50 to 100 of these bubbles. So quantifying them and determining where they're located is a little bit difficult. So we're combining our tools with machine learning and, things like microfluidic systems that we use as well to handle our animals to try to understand better how these beats are formed. And more importantly, what we really want to understand is, do they have any functional meaning? It's possible that these neurons are just changing shape, but they still work just as fine. We don't think that's the case, but to show that there's actually significance to these changes in shape and morphology, we want to know what about their changes in shape actually are related to changes in function. And the reason to do that is because then we could have a target for treatment. If we realize that it's actually the beats that are inducing changes in function, or if it's something like breaks in their dendrites or wavy processes, all of these are things that show up with aging. Which of these features actually represent a pathological feature? That's what we're really trying to understand, so that we can then try to figure out how can we fix it or prevent it as animals age.
Definitely. And I know studying aging in humans is a very long process. We're talking decades and decades here. For worms, what sort of timescale are you working on?
So that's something that's really great about C. elegans. They can live anywhere from 2 to 3 weeks. So it's super fast in comparison. You can also do a lot of different modifications and tests to see what happens. You can expose them to different drugs. You can expose them to different environmental conditions, or you can induce different genetic changes. and see what's their effect. And you can do this in very large scale, right? In comparison to what you could do with higher model organisms, right? So it's faster and it lets you just test more perturbations than in other systems. So that's one of the very good advantages of working with C. elegans.
Well, Adriana, this project is amazing, and I'm so excited about the work that you're doing, but I know it's not always easy. As we alluded to, in science, there are challenges, there are struggles, and lots of failures. And we love talking about these on our show because I think it's so important for trainees out there to discover that they are not the only ones having these struggles and failures. So for you, do you have an example from your own experiences of a time where everything was just going wrong, or you had a major failure that you had to work through that you could share with us?
Many. To be honest, I think the students that don't encounter those failures are really the outliers, right? And I always tell my students, if you're not encountering these things, that means it's because you're probably doing something that's already been done and we already know how to do it, right? So it's part of the process. I have many instances where things have failed. And for example, in my PhD, we were trying to make these little capsules that will release their contents upon a very, very mild change in pH conditions. And I had been working on trying to make these little capsules for probably a year. I don't even remember how long, but it just seemed like a very long time. And I tried many, many different things and nothing worked, right? So I was ready to give up and I went to meet my advisor and I just said, I think I need to change projects because this is just really not working. I don't see how these can become something productive. And that just tells you I had failed a lot of times. And he said, well, you know what? Let's just give it one more try, just one more try. And if this one more thing doesn't work, then we'll change it, right? And that one more thing was something that we had talked about, but we hadn't tested. And of course, that one time it worked. And that became sort of the bulk of my PhD project. And I produced multiple papers after that. And it's one of probably my highest cited papers at this point. So there's many stories like that. I think for my postdoc, you think, oh, once you graduate, this is over. No, in my postdoc, I also encounter multiple instances of things like this and where you have to work through it, repeat experiments multiple times. But at the end, I think persistence is very important. And not taking it personally. It's not you. That's just how science is done. And that's the nature of the process.
Absolutely. And I love the story where you were just on the verge of giving up on something and then you had a breakthrough. And I think those moments are just some of the most memorable and rewarding moments in science because you had to work so hard for it.
That's right. Yes.
Well, we don't just want to talk about the tough times. Adriana, do you have a favorite success story you'd like to share, whether it was a big win or even just a small victory recently?
Yes, I think recently I'm very proud of our most recent paper that we published. And probably the reason I'm very proud of that is because that was a difficult project. And I think the students that worked on that had to work very hard. They were one of my first PhD students and it wasn't a very easy project. I tried multiple things before doing this and eventually we got it to work. We actually found something very interesting. So just to give you sort of a super brief description of what it is, we were trying to find ways or new methods to identify genes that are relevant to aging. And we tried multiple ways of doing this. And at the end, after many failures, we found a new marker that we thought we could use this as a marker for aging. And this marker is stress granules. So these are small liquid condensates in cells that form when there's a stressful environment, but they also form when worms age. And so we said, well, we can use this formation of these little stress granules as a marker for aging. And if we find animals that have a premature formation of these stress granules, they probably are going to age faster. And so that can tell us which genes are changed in this particular animal. And so we use that marker and we combine that with our image processing and microfluidics tools. And we actually found some interesting results. And then after corroborating our analysis, we actually found an interesting gene that we're actually starting to work on now that people have not associated with aging before. And we think it can be very relevant to identify new pathways in the aging process. So that was this new paper, and we're very proud of that one because that one was a hard one, but we actually got some very nice results from it.
That is fantastic. Well, congratulations to you and the team there in the lab.
Thank you.
So how did you celebrate this exciting success?
We had an outing with my group. We decided to celebrate when we submitted the revisions to the paper, because I always think that's a point where I like to celebrate, because it's kind of saying all the hard work we did, we deserve to celebrate this. And everything after this is out of our control, even reviewers like it or not. So I like to celebrate at that point. And we all went out and got dinner together.
I love that. And I completely support having a lot of sort of provisional celebrations along the way. So
as many as possible.
Well, that is wonderful and exciting success for your laboratory. And I think it is so important to take a moment to celebrate these wins, because research can easily consume you. It's easy to be just wrapped up in the literature, reading everything, working all the time. But I always recommend for our listeners to take a break, to give their mind a break, and to read something for fun. So I love giving everybody book recommendations on the show. Adriana, do you have a favorite book, whether it's related to science or not, that you can recommend for all of us today?
Well, I have many books that I like. I read mainly literature. A book I read recently that's kind of sciencey. It's called Why Fish Don't Exist. I really enjoyed that book. It's part of a story of how we like to classify organisms, right? And how that's sort of a human mind construct. So I really like that idea. And there's a lot of history behind how we got to that point and the first people that were focused on classifying different types of fish. But then there's a lot of personal stories intermingled with that. So it's just a beautiful book. I really liked it.
Wonderful. Well, you are, I believe, the first to recommend this. So we will add Why Fish Don't Exist to our website for our listeners to find there. Thank you so much for that recommendation. And we've talked about some different aspects of your career, but one of the things I love to emphasize about careers in science are the opportunities to travel. You get to go all over the world for meetings, to meet with colleagues, to work with collaborators. So Adriana, do you have a favorite place that your science has taken you?
I really enjoy traveling to the C. elegans meeting, which used to be in Los Angeles every 2 years. And now for the first time, it's going to start rotating. So it's going to be in Scotland this coming year. So I'm excited about that. Hopefully I can make it. There's also the other meeting I usually go to is the chemical engineers meeting. And that also tends to rotate throughout the US. So I like the opportunity to be able to go to different places.
Well, this meeting in LA sounds fun. And I know going to the same place every year, every 2 years, and being able to kind of reconnect with colleagues is an amazing experience. Can you talk about what it's like when you go to these regular meetings?
Well, it's really nice to be together in a room with people that sort of are really in your same field, people that whose papers you've read online, right? Or you know about the work, and then you see them in person and you just start interacting. People in the C. elegans community are extremely nice also. So it's a really fun meeting to go to and to see old friends and to meet new people and new students. It's really nice.
Wonderful. And have you been able to see some of the sites around LA?
Not so far, but maybe there will be another opportunity to go there. But I'm excited about the potential of going to Scotland next. So maybe I'll be able to visit there.
Will this be your first trip to Scotland?
Actually, no. I was there when I was young. I did a study abroad experience in my undergrad. And so I went to England and I was able to travel to Scotland. But it's been a long, long time. And so maybe it's time I go back.
Well, we'll keep our fingers crossed that you are able to make it to this wonderful meeting in Scotland. And it sounds like you've had some great opportunities to meet with colleagues, to go to these conferences across the United States. And I think one of the things that makes science so great are the people that you get to work with and interact with. And I think oftentimes there's this sort of stereotype about what scientists are like and what life as a scientist is like. And I found that in many cases is just not true. There are so many amazing, creative, funny people in science. And it sounds like you've got sort of a collaborative, warm environment there. So what is it like in your lab? And do you guys get together a little bit outside of the kind of usual 9-to-5 science?
Oh, yes, absolutely. I think for me, when I started my group, that's something that I said from the get-go. This is important to me, having a collegial and just a nice, friendly environment. Because science can be hard, right? And just dealing with experimental failures multiple times, it's just hard, right? And I think having colleagues that become your friends to just be with you at that time, it's very important. So I really try to nurture a collegial environment in my group. I think that we have a really good culture, and that's stayed throughout the years. I think that's something that I tell people when they're going to join my group, just go and talk to the rest of the group. And I get feedback from everybody. And we make sure that the new person that's coming, I tell them, you're going to be working with these other people and just hanging out with them, right? So you want to make sure everybody likes each other.
Right.
Which is most of the times is the case, right? But we also have a lot of outings as a group that help build that sense of community. This semester, for instance, we started over the summer. We did this really, really fun hike all together. Here in North Carolina, we have these really great greenways and hiking trails. So we all went together for a hike that we really enjoyed. And then we usually have celebrations for things like papers, submissions, promotions, anything like that. And so we actually have our holiday party on Thursday. So that'll be the last one for the year.
Oh, that's wonderful. So do you have any particular traditions, things that always happen at the holiday party?
We don't have any particular traditions. People are welcome to bring their ugly sweaters. Maybe about half of the people do that. But we just get together, have dinner, and just have fun.
I love that. Do you have a favorite ugly sweater that you wear?
Not really, no.
No, you got to work on that, Adriana. You need one with like elegans and Santa hats. And
That's right.
Well, that sounds wonderful. And I think having this warm, welcoming collegial lab environment is so important for science, because I think you can get so much more done when people are working together than when everyone's sort of in their own corner of the lab working independently.
Right. Students are here for like 5 years average.
Mm-hmm.
You want to make sure that that time is still part of your life, right? You want to make sure that it's a time in their life that they're enjoying as much as we can make that happen. So
absolutely. And you are answering some amazing questions and solving some big problems in the lab. And I know oftentimes scientists are limited by unfortunate things like funding and staff and time and the feasibility of the project. So Adriana, if we took away all of the barriers that normally hold you back and gave you everything you could dream of, what is the one problem or question that you would want to tackle first?
That would probably be neurodegeneration. I mean, I think neurodegenerative diseases are just really, really tough. And with an aging population and aging being the main risk factor for these neurodegenerative diseases, this is just going to become more and more common. And after years and years and years and a lot of hard work from a lot of scientists, we still don't have solutions for many of these problems. So I think that's something that really, really motivates me. I'm just completely terrified of encountering any of these diseases in myself or somebody in my family. And it's just something that really motivates me. But yes, it's very, very difficult. So if I had all the money in the world and all the resources, I would probably put all of those resources into that.
Absolutely. I think particularly as we've got this sort of aging global population, neurodegenerative diseases and some of these just other diseases of aging become even more bigger public health problems. So this is a worthy cause for unlimited resources. And we've covered a lot of different topics in our conversation today. Adriana, I'd love to end by talking about advice. So for you during your own career, during your own life, was there one particular piece of advice that somebody gave you that really helped you that you can pay forward to our listeners today?
I think, and I kind of mentioned it before, but at some point during my postdoc, I was deciding on whether I would take a second postdoc in a different place or not. And there were some family considerations that came into play as well. And I was trying to get advice from multiple people. And somebody I got advice from was a professor at Georgia Tech. And he basically said this to me, which is there's no direct path. Just knowing that if you have multiple options, there's no right or wrong answer. If you take option A or B, it might change your path somewhat, but there's no right path to get to where you want to go. You just look at your options and go with what you want or makes sense at the time. I think that's important without maybe stressing out too much about the long-term effects that that's going to have, because most likely you will be fine with either option. And the other thing is really also that you don't necessarily need to know from the day you enter undergrad what you want to do with your life. You don't need to know if you want to be an academic or whatever. I think it's also a combination of what you want, what you're good at, what you enjoy, and what opportunities are out there and what the world is looking like. So at least I think, was very stressed about that. Like, where do I go? What should I do? And I think looking back, it's most likely that you will be okay and that the choices you make are going to lead you to do something that you like and enjoy. And you don't need to know at the very day of your undergrad, what's your ultimate goal? It's okay to figure it out as you go.
Well, Adriana, I'm so glad you shared this because I think it is easy to just agonize over making every decision and feeling like you have to have every step of your career planned out. And there's so much pressure, I think, on students these days. But I think you're absolutely right. You just sort of go with the flow and take those opportunities as they arise in front of you. You might discover something completely new that you're very passionate about and excited about that leads you to a different direction that is equally wonderful.
Definitely. I think there's things you maybe don't even know about, and then an opportunity will come up and then you'll realize, oh, that's what I want.
Well, great advice for our listeners. Is there any other last piece of advice or maybe a last note of inspiration that you'd like to leave everybody with at the end of our conversation today?
Another thing would be to try to look at the big picture of things, because sometimes we get very focused on one thing that's not working or very, very stressed about one thing, but maybe taking a step back. And I think the pandemic kind of tried to push us to do this a little bit, but taking a step back and getting a little bit of perspective on overall, are you okay? You're going to the right direction. And students and everybody's really stressed right now. So I think taking a little bit of a bigger picture perspective can help, knowing that you're all right and you're on the right track. I think that's important too.
Absolutely. Well, wonderful message to share with our listeners. If they would like to learn more about you, Adriana, and the work that you're doing, what is the best way for them to do so?
Probably my website. So sanmiguel.wordpress.ncsu.edu. You can probably also find it if you Google San Miguel Lab at NC State.
Perfect. Well, listeners, definitely check out that lab website. Learn more about the amazing research that Adriana and her team are doing. And Adriana, it's been such a pleasure to chat with you today. Thank you so much for your time.
Likewise. Thank you so much for the invitation. I really enjoyed it.
Well, we've enjoyed spending time with you today. Listeners, great to have you here with us. We look forward to seeing you in our next episode of People Behind the Science.